We are deeply grateful to Dr. Shairul Izan Ramlee. Welcome. She is from University Putra, Malaysia, for joining us as our distinguished lecturer. Your expertise and dedication will undoubtedly enrich our understanding of molecular markers and their crucial role in germplasm characterization, genetic diversity analysis, and conservation strategies for sustainable crop improvement. And also to Ms. Octaviani Karu, thank you for your willingness to be the moderator who will oversee this event until it ends. I heard that this lecture will be conducted in two sessions. Session 1 in June 15 and session 2 in June 17. And I'm sure that this event is not only an academic forum, but also a bridge that strengthens the partnership between Universitas Brawijaya and Universiti Putra Malaysia, fostering a spirit of scientific cooperation that transcend national boundaries. I also encourage all participants to actively engage, exchange ideas, and take full advantage of this opportunity to broaden perspectives. I'm sorry. We hope that this lecture inspire us to continue developing innovative solutions for the challenges facing agriculture and biodiversity conservation in 21st century. Finally, on behalf of the Department of Agriculture Science, Faculty of Agriculture, Bioindustry, and Forestry, I extend my warmest welcome and wish you all a fruitful and enlightening experience. Thank you very much once again. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much, Dr. Darman Saptadi, SPMP, for the warm welcome and inspiring remarks. And now we shall proceed with the highlight of today's event, which is the guest lecture session. So,
Okay, thank you, our moderator. Okay, Assalamualaikum and welcome everyone. Good afternoon. How's everyone today? I hope everyone ready for the lecture. Okay, so anyway, there is anyone that can share the screen for my slide?
okay, Doctor, I will share screen the material.
Okay.
Wait.
Okay. So, so today we are going to explore the pillar that driving the crop improvement in 21st century with the title Molecular Markers for Germplasm Characterization and also Genetic Diversity Analysis. Okay, we move on to the next slide. Okay, currently we are facing global crisis with the food security due to the climate changes and also the growing population. Okay, in order to answer to this crisis, okay, we lies in the foundational bedrock of all breeding program, which is our germplasm. Okay, so germplasm refer to the genetic material of plants that we use in breeding, conservation, and also research. These include everything from the from the current cultivar that we actively using in modern agriculture, to the traditional land races, adapted to the hyperlocal environments, and also even wild ancestral relative, okay, that holds vast untapped genetic potential. Okay, so today there are more than 750,000 global accessions stored in the gene bank around the world, with the massive backup repository like the Svalbard Seed Vault holding with over 1.3 million seeds. Okay, this is in order for to protect our global food security. But here is the critical problem. Okay, having those seeds stored in a frozen vault is not enough. Okay, if we do not understand the genetic inside those seeds, they are actually useless to us. Okay, next slide. So this brings us to the process of germplasm characterization. So without thorough characterization, those thousands of accessions remains nothing more than the unutilized black box. Our primary goal in characterization is actually to open those boxes and to identify the specific gene related to desirable trait like heat tolerant, drought resistant, disease resistant, and also to increase the yield and convert the raw genetic material into actioning breeding information. Okay, so in the characterization, we use this workflow that begins with the initial screening during the pre-storage and also the collection, followed by the detailed post-acquisition evaluation in the field, and also strategically prioritization so we can optimize our resources. Okay, next slide. When speaking about the characterization, we basically mostly rely on these four major type of characterization. So I think most of us familiar with the morphological characterization, which evaluate the visible agriculture traits, okay, like plant height, grain size, okay. And then we also have the physiological characterization where we assess the functional traits like water use efficiency, okay? And we also have a biochemical which involve analyze seed protein and also ILO enzyme, okay? And these actually serve a vital bridge, okay, between the physical phenotype and the underlying genotype. Finally, we have reached the modern standard, our molecular characterization that allow us to directly analyze DNA to identify the genomic variations. Okay, we go to the next slide. Okay, so basically here the slide show the comparison of about morphological versus molecular characterization. Okay, so let's compare the traditional morphological approach with the molecular approach. Okay, so molecular characterization is basically, so this is actually the slide show only the summary. So morphological characterization is highly influenced by the environment, and that often require us to wait until the plant reach full maturity. Okay. However, okay, for molecular characterization, okay, is actually a game changer. So basically in molecular characterization, it looks the direct DNA sequence variation. Okay, like the variation that we talk about is like insertion, deletion, and also mutation. So making this completely unaffected by external environmental. So it can be applied at any stage or even when the plant is just a seedling. So that is the benefit of molecular characterization versus morphological characterization. So we go to the next slide. So when we talk about molecular characterization, of course, we use molecular markers. Okay. So basically, what makes an ideal molecular markers? So molecular markers act as a precise genomic signpost. Okay. So basically, there are variation in the DNA sequences, such as insertion, deletion, and also mutation at the genomic level. And to make this can be used as a molecular markers, it needs to be highly polymorphic, meaning it can detect multiple allele at a single genomic locus, okay? And it must also be a codominant markers. This allow us to distinguish between the homozygous and also heterozygous plant, okay? And another criteria for the molecular markers that it must also be highly reproducible, okay? And also abundance across the whole genome, okay? And this is to ensure that our diversity data is unbiased, okay? When we use all markers with these properties, like the highly polymorphic, codominant, and also reproducible abundance, the efficiency gain can be improved. Okay, we can predict phenotype without massive field trial. Okay, reducing time that require for a breeding cycle from 8 to 10 years down to only maybe 3 to 5 years. Okay, and also it can allow us to optimize our gene bank by identifying the core collection. Okay, so core collection is actually representing the maximum genetic diversity of a species, okay, using only 10 or 20% of the store accession. Okay, so obviously this can save on the labor and also the preservation cost. Okay, so we move on to the next slide. Okay, so now we already understand what are the properties or what are the fundamental of an molecular markers. Let's look at exactly how this tool actively support and transform our operation in the breeding program. Okay, so basically, I will break down this impact into three major pillars. Okay, so the first pillar is the selection of the parent. Okay, so in traditional breeding, okay, selecting which parent to cross can sometimes be a guessing guess. Okay, based purely on what they look in the field. Okay, however, when we are using molecular marker, we can strategically identify, okay, genetically diverse parent at the DNA level before they even flower. Okay, so by doing this, okay, we maximize what we call transgressive segregation. Okay, so the phenomenon, I hope the terms everyone familiar with. Transgressive segregation is the phenomenon where the offspring basically outperform both of their parents. Okay, and so by also using marker data, this also can prevent us, okay, that from accidentally crossing close related plants, okay, therefore avoiding genetic bottlenecks. Okay, so that is also the use of molecular markers in order for plant breeder to select the parent. Okay, the second one is the breeding efficiency. Okay, this provide the most immediate economic benefit to a breeder. Okay, because molecular marker act as a direct genomic signpost, so we can accurately predict a plant future physical. Okay, simply by reading their DNA. Okay, so these actually drastically can reduce our reliance on the massive and also expensive field trial. And okay, by implementing technique like marker assisted selection, genomic selection, okay, we can actually shorten the breeding cycle. Okay, so which traditionally took 8 or 10 years of growing and selecting, and now can be compressed only 3 to 5 years only. Okay, so our third pillar involve the conservation management. As we discussed earlier, right? Okay, we have more than 750,000 station sitting in the gene bank, okay, globally. Okay, and this is impossible for us, plant breeder, like the worldwide, okay, to evaluate all of them in the field. Okay, so the role of molecular marker here allow us to optimize the management of this massive repository, okay, by identify what we call the core collection. Okay, this means, okay, we can capture and represent the maximum genetic diversity of the entire species by only using 10 or 20% of the store section. Okay, and finally we have the four pillar that cover the advanced application of molecular markers. Okay, and that is advanced application. Okay, so instead of just making crosses, okay, marker can be used as tools, okay, for plant breeder. Okay, they can serve as they are essential for QTL mapping that allow us to trace phylogenetically and also the evolutionary history of the crop. Okay, and also they can also be used to do the purity testing, okay, such as in the hybrid breeding seed production. So we can have those pure hybrid seed. And so basically these four pillar show that molecular markers is actually the foundation of the precision breeding era. Okay, so we move on to the next slide. So can we move on to the next slide? Okay, so to appreciate molecular marker, we must look at the history of how this marker technology evolved. Okay, so basically, we have one generation, first generation, second generation, and also the third generation. So our first generation tool is actually is a hybridization based marker, which is specifically we call as a RFLP. Okay, so this type of marker has been pioneered in the 1970s and also 80s to build the first genetic linkage map. Okay, so the foundation works of these markers is basically they cutting the DNA with the restriction enzyme and then use the Southern blotting to detect the variation fragment. Okay, so while they are actually highly reproducible and also they are codominant markers, they are actually slow and also resource intensive and require radioactive probes. And then next slide. So after the hybridization markers, okay, next came the PCR-based marker, okay, like RAPD and also AFLP, okay. So these two marker is actually the improvement of the RFLP markers, okay. So RAPD use a short random primers, okay, so making them very cheap and also simple, okay, but they suffer from the poor reproducibility and also dominant inheritance. Okay, So meaning that they cannot distinguish the heterozygosity. Okay, and then AFLP is combined restation digestion with a PCR is actually the highly reproducible markers, high throughput, okay, but actually they remain technically demanding. So you need a lot of PCR, okay, amplification to do this. Okay, next slide. And then today, okay, so nowadays we often use advanced PCR marker such as SSR and also SNP. Okay, so SSR stand for the simple secret repeat. Okay, or that detect variation in the tandem repeats. Okay, the SSR actually is the scattered throughout the genome. Okay, they are codominant. They are highly polymorphic. Okay, and we always use this as a marker of choice to do characterization. Okay, though they require prior knowledge about the sequence knowledge. Okay, but the true model standard is actually SNP markers. Okay, or single nucleotide polymorphism. Okay, or SNP. So SNP detect the individual base pair. Okay, because in one genome, okay, we can have like million of SNP, okay, per genome. Okay, so these can be processes and also can be used to as molecular markers, and then making them the ultimate tool to do a genome-wide association studies or genomic selection. Okay, next slide. So now we already know about the markers, like we have the AFLP, we have SSR, we have SNP, okay? So now whether we are using SSR or also SNP, we must also evaluate the quality of the markers, okay? So what actually make a marker truly informative, okay? So it comes down with these two. Okay, so high allele number and also a balanced frequency. Okay, so we need to have a high number of distinct variants. Okay, to increase the power of the markers, and also, however, being only having many allele is also useless if the frequency is unbalanced. Okay, so we need to find markers that have high allele number and also beside with the balanced frequency. Okay, so next slide. So this is where the concept of polymorphism information content, okay, or PIC we call become crucial. Okay, so for any given number of allele or even a uniform frequency distribution, the yield highest possible gene diversity and also expected heterozygosity. Okay, so if a marker distribute is heavily skewed, okay, meaning it is only dominated by one or two allele, okay, while the other are rare, okay, this create a weak signal. Okay, so these suboptimal marker are actually vulnerable to genetic drift. Okay, meaning those rare allele is actually not useful. Okay, so high informative marker with uniform distribution provide a strong signal. Okay, so by maximizing the PIC value, okay, we can also enhance the power to do a genotype discrimination, okay, especially in the large scale population. So this type of polymorphism analysis is incredibly reliable. Okay, so next slide. So this is actually the summary table, okay, of from all those conceptual and also the mathematical principle together. This is you can use this table to make a rapid decision in the laboratory. Okay, rely on the marker selection diagnostic matrix here. Okay, so basically this matrix act as our checklist. Okay, drawing clear line between the markers, which are the marker that can help us, or which are the marker that can lead us to a weak signal or misleading data. Okay, so the first row of the matrix is the first row of this matrix is the frequency distribution. Okay, so as we establish the highly informative marker, okay, that display even and also uniform across each allele. Okay. On the other hand, the suboptimal marker is actually the skew and unbalanced markers. Okay. Meaning the marker is completely dominated by one or two highly common allele. Okay. So this fundamental imbalance is the root cause of the poor marker performance. Okay. And then the second criteria is the PIC, okay, polymorphism information content. And so basically if the highly informative uniform distributed marker will naturally, okay, maximize the gene diversity for the given number of allele. Okay, in contrast, okay, for suboptimal marker, so severely limited by the skew, okay, such as dominant allele. Simply put, the overwhelming presence of one common allele, okay, drown out all the genetic signal of all of the others. Okay, so unbalanced distribution of dominant alleles. Okay. So what if we happen to apply this marker? This bring to our third criteria is actually the genetic drift susceptibility. Okay, so highly informative marker is actually incredibly resilient. Okay, so even you are working with smaller population sample size, okay, which is common, okay, in early stage of breeding trial. And even distribution can ensure your marker remains reliable. Okay. However, in the suboptimal marker, they are actually highly vulnerable. Okay, because their alternative allele actually exists in a low frequency. Okay, and they are actually susceptible to the genetic drift. Okay. So in a small sample size, those rare alleles might simply not be drawn, meaning they frequently are presented or permanently lost in the data pool. Okay, so then we reach our practical application suitability. When your marker check all the boxes, okay, on the left side of this matrix, okay, the result is weak, and also you will have a poor discriminatory power. Okay, you will you will struggle to tell the closely related plant, okay, undermining your entire conservation and also breeding effort. So before you want to run the marker, so I hope you can go to refer to this table and see where is your marker. Okay, so if you have the marker that that tick all the boxes for the highly informative marker, you can carry on with the markers. And if they are not, okay, usually you should stop and then maybe proceed to the next marker. Okay, so next slide. So basically, okay, after all this data from the marker, okay, then we will have the what we call a reliable data, okay, we can perform all the analysis, okay, for the diversity and also the population analysis, okay. So these are the analysis, one of, I give this three example of analysis that you can use, okay. We can use to do a principal component analysis, PCA. So basically PCA is helping you to visualize, okay, your genetic variation, and also we can use this to detect the redundant genotype, okay. So the second one is the cluster analysis, such as the UPGMA method, okay. So basically using this method, they group the accession based on the genetic similarity, and also then they will produce a phylogenetic relationship, okay. And next to it, we also have a neighbor joining analysis, okay. So it's also do the same, only for the UPGMA is actually apa, we use a constant evolutionary trait, so making, if you have more closely related accession, you should use this UPGMA method compared to neighbor joining method, okay. So I also list out what are the software that provide those analysis, okay. And we also have the model-based Bayesian clustering tools, okay, to do population structure analysis, okay. So it looks to on more on the population level, okay. So like genetic ancestry, hybridization, the integration dynamics, okay. So you can use this kind of analysis. Okay, next slide. Okay, so basically in this slide, okay, I put the case study for all the not all, okay, some of the case study that use molecular markers in their research, okay. So this is the first one is 3000 rice genome project, okay. So this is a global known project, okay, we call as 3K rice genome project. So basically this research is using, okay, 3000 accession from 89 countries, okay. So they are resequencing back all the accession, okay, and then they map back, okay, and align back with the reference genome. And after the realigning, they discover about 18 million SNP. Okay, so after that, okay, after that they use those marker, and then to group this accession together. So out of the 18 million SNP, okay, they divided all the 3000 accession into 5 major varietal groups, okay. So this basically providing our world largest genomic database for climate resilient rice breeding. Okay, next slide. So another case study is for the oil palm architecture. So basically this is from a research from my student. Okay, so we published this last year. Okay, we call as a genome associate study. Okay, in oil palm. So the study focused on oil palm architecture. Okay, so we utilize GWAS. Okay, to target the compact plant ideotype. Okay, in order to facilitate easier mechanical harvesting. Okay, so basically we genotype all 475 palm, okay, and in the customized panel diversity of 5,000 SNP. Okay, so out from the 5,000 SNP, okay, so we found that 27 specific marker that strongly related to plant architecture. Okay, apa tu? Specifically on the compactness of the trees. Okay. So these 27 specific markers can be used as universal markers for all palm, okay, or selecting all palm for this trait, okay. So this can basically help serve as the foundation for marker assisted selection that will also reduce the labor cost and also field trial in all palm. So next slide. Okay, next slide is also one of the work of one of my student. Okay, so basically we study on the eggplant variability. Okay, so we combine all set of morphological trait, okay, with DNA markers, okay, and we evaluate 29 different eggplant accession under different cropping condition, okay. So from the cluster analysis, we are able to group this accession, okay. So basically, we can allow reader to map out specific crossing between a distinct group to achieve maximum heterosis or hybrid vigor, providing a precise roadmap for local crop improvement. Okay, next slide. And beside those three case study that we already show just now, okay. So molecular markers can also be used to to improve the or integrate in the advanced breeding and disease resistant objective. Okay, so for example, using marker assisted back crossing. Okay, so here, breeder can precisely integrate the specific disease-resistant gene from a donor plant into an elite cultivar. Okay, so in our research, okay, we discover that 96% of desired desired parent genome, okay, we can achieve. Okay, we can recover like around 97% of the parent genome in just 5 generations. Okay, with the use of marker assisted back crossing. Okay, and another one, we can also perform a gene pyramiding. Okay, so where we stacking multiple resistant allele together. Okay, like 4 different blast resistant gene in rice. Okay, this is to provide a durable resistance or also introduce a multi-layer defense against the pathogen. Okay. And finally, we also use GWAS, okay, to correlate high density SNP marker, okay, such as the oil palm work just now. Okay, so next slide. So looking at the future, actually, we actually are in the midst of the next generation sequencing revolution. Okay, so before this, okay, for next generation sequencing, the cost is definitely too high. However, these are going down under like $500 by today. Okay, so the cost decline is allowing us, okay, to have more sequence that are under study before. Okay, and we can also apply to the orphan crop. Okay, crop that we are not focusing. Okay, or that is what we call as a orphan crop. Okay, and we also are moving beyond just DNA into a multi-omic. Okay, so basically not only DNA, not only on the molecular side, but we also want to add on the transcriptomic data to understand the gene expression and also metabolic to profile like a nutritional quality. Okay, and digital transformation, introducing machine learning model that can predict the performance straight from the genomic data, okay, before we start the field trial. Okay. However, basically we have to acknowledge, okay, that we still face a significant bioinformatic gap, especially in the developing nation such as Malaysia, okay, Indonesia, and then we also have complex legal issue regarding the IP right, okay, and also obviously to have the initial the cost, okay, initial cost for the infrastructure is also high, okay. That is also one of the challenge for molecular markers to be implemented, okay, in our nation breeding program, okay. And I think this is my last slide, okay. So basically in conclusion, okay, by understanding the genetic diversity is actually what the ultimate foundation of our future food security, okay. So the challenge of climate change, population growing are vast, okay. But as we seen today, okay, molecular markers serve as our most essential lens, okay. So they provide us to identify, preserve, and how can we strategically deploy the exact heritable trait that we need to build a sustainable and also climate resilient agriculture future. Okay, with that, thank you, and I'm open for more question from all of you. Thank you.
Okay, thank you very much, Dr. Shairul, for the insightful presentation. We will now proceed to the session of question and discussion. To all the participants who would like to have a question to Dr. Shairul, please raise your hand, or maybe you can just drop your question in the chat box, and please kindly mention your name. Is there any question from the audience?
So everybody understand what the lectures today?
Okay, maybe while we wait for the question from the participants, perhaps I could start with my own question, Dr. Izan, if I may.
Yeah.
So, based on your experience, what do you think is the biggest challenge to integrating the molecular markers into the practical breeding programs, especially maybe in, like you said before, in the Malaysia or maybe in developing country like in Indonesia?
Okay, like I said, okay, I would say the biggest challenge for developing country like us, okay, is actually the cost, okay. Sometimes the grant that we got from the ministry is not that big, even though the sequencing rate is actually going decrease, okay, now, but it's actually is still a lot, okay, especially when we are working in really big sample or in big population, because in plant breeding we are not only doing one or two individual, okay, we work on the population level. So that is then if one sample costs us like $150, and then you have to times like if you have 40 individual in your population, then you have to times 40, right? So that is also big challenges, okay? And also that is regarding the facilities as well, okay? So I'm not sure if not all university in Malaysia at least, okay, have the capacity or the facilities that we can do a sequencing on their own, okay. So like us, we always outsources them, okay. And then when we outsources them, and then we also have difficulty on the bioinformatic expert, okay. So not all are familiar with how to analyze the data and how can we generate those data into something that are biological, that have the biological meaning from the because the data that you will get is something like the best pair, right? Like ABs, not CGAT and blah blah blah, right? So not not everyone are familiar how to at least analyze those data into a meaningful explanation, okay? The science behind those data. I hope I answer your question.
Okay, thank you very much, Dr. Izan. Jadi, maybe it's more like the cost and the facility out there, right?
Yeah, I think that is also one of a big issue, because if you want to go into a deeper, like the fundamental out of that, people can learn. Okay. So but however, if those people, they are not equipped by all those facilities and also all the resources that they need, especially the software. Okay. Because in my presentation, okay, when we want to do the analysis, we need to use all those software. Okay, so some of the software are not freely available. Okay, so that is also, I think, one of the big challenges, especially the student. Okay, the plant breeding student or agriculture student that don't have the access to those resources.
Okay, thank you very much. That's actually your answer my question. So maybe, is there any questions from the other audience? You can just raise your hands and mention your name and also deliver the question. Okay, maybe first from Rebel Brevo, please, your question.
Thank you for the time. I want to box about something. I am still confused about the difference between germplasm, excision, and landrace. Could these three terms be explained by explaining using a simple analogy?
Again? Can you repeat the question?
All right. I am still confused about the difference between germplasm, excision, and landrace. Could these three terms be explained using a simple analogy? Something like that.
Oh, okay. So, thank you for your question. How do I want to call your names? Bravo, is it? Okay, so basically for land races is actually uh the variety is actually locally adapted by the farmers. Okay, so it's being traditionally developed by generation by the farmer. Okay, so however, okay, accession is a distinct and also unique different sample of plant materials such as it can be as a seed, tubers, okay, that are officially adopted and maintained within the germplasm collection or the gene bank. Okay, so basically, if you have the accession, right? So we have the rice accession, okay, store in the ERI gene bank. Okay, so that is what we call accession. However, land races is actually the seed that that developed by the farmers, that they do the by generation, that are locally adapted. So maybe land races is uh, uh, if the farmer living in the highland of the country, so maybe the seed that they develop is have the traits for highland soil. Okay, that is also one of example. Okay, so basically land races is already adapted and also evolved through the natural selection. Okay, such as the local soil, climate, okay, and also maybe the area of the farmer live, okay, because the farmer usually they save seed from the plant, okay, because of the prefer the taste, okay, or they look that, oh, this plant have a very high sweet sugar content, okay, so that is why they save the seed for the next season, okay, so those seed is actually land races.
Sorry, is there any feedback or the answer is enough?
Enough, yes. Thank you for the answer.
Okay, thank you so much, Dr. Izan, for the answer. And maybe next question from Kaulin. Please deliver your question.
Okay, thank you for the opportunity. Okay, I will ask question for regarding the eggplant case study. From the study, it's mentioned that breeders can achieve higher heterosis by by crossing accession 1 with group 1, group 4, 5, or 6. Also, and my question is from a genetic standpoint, is there any potential risk of cross incompatibility if the genetic distance between this group is actually too wide or too narrow? Thank you very much.
Okay, that you refer to which case study there?
Eggplant case study.
Eggplant, okay.
Yeah.
So basically those the case study for the eggplant, wait, I to open it here. Okay, so basically, okay, here usually what we do, okay, when we have those, like, this is what we call as we do a clustering, right? So basically we group all those into few groups. Okay, okay, so we have group 1, group 2, group 3, 4, and 5 and 6. Okay, so basically from this we can actually find the how related those accession, or how related between the sample between each other. Okay, so if you want to have hybrid heterosis, okay, usually heterosis we use from bigger distance, okay, of the group. Okay, so the bigger is better. So that is also one of how we can translate this into some plant breeding decision. Okay, so basically we can try to do because, okay, I cannot guarantee, okay? Of course, we plant breeder, we cannot guarantee, but this the phylogenetic tree serve as a foundation for us to decide what are the best decision in our breeding program. Okay, so I will not it will not guarantee you to get the heterosis. Okay, however, okay, from the technically, okay, if how we want to achieve heterosis, we should use a distant parent. Okay, but the more distant, then we can get more heterosis because we introduce new gene pool, right? So that is the foundation of the heterosis. So using that, okay, we select the parent out from a few cluster. Okay, maybe from cluster 1, maybe we select 2 parent. Cluster 2, we select another 2 parents. Okay, and then we do the crossing out of them. Okay, so I will not say that we can guarantee you that we get the heterosis. However, usually we can do it. We can get it based on the fundamental from heterosis itself. That if you use a different or distant group, okay, of parents, we can possibly and ensure that we have high probability to get the heterosis.
Thank you, Miss Shairul.
Thank you. So it will not guarantee you, but it can serve you it can serve you as a foundation for you to decide, okay, or to do your breeding decision. Okay.
Okay, thank you so much, Dr. Izan, for the answer. And I think before Mustafa has raised hand, or maybe Mustafa, do you have questions?
Uh, yeah, Assalamu alaikum. Good afternoon.
Good afternoon.
Thank you so much, Doctor, for your insightful. And I am Mustafa Jadao, an international student from Africa, Gambia precisely.
Mm-hmm.
I want to ask a question regarding from the context of germplasm characterization. How do you determine the optimal balance between the high throughput SNPs markers and more traditional SSR markers for maximizing genetic diversity detection and breeding efficiency, especially when considering resource constraints and the need for robust population structure analysis? So, uh, I just wonder from any recent studies that
Um, Mustafa, actually, I didn't get all of your question because your voice is flickering. So maybe maybe our moderator can help us? You asking
yeah,
about it?
I'm using earpiece, so I don't know whether the network. I hope you are hearing me now.
Yeah,
Yes, Mustafa, can you repeat your questions?
Hearing me now.
Yes, it's clear now.
Okay, I am from the context of germplasm characterization. I just want to ask her how can she determine the optimal balance between using a high throughput SNPs markers and more traditional SSR, because we have SNPs and traditional SSR markers for maximizing genetic diversity detection and breeding efficiency, especially when considering resources constraints and the need for robust population structure analysis. So I'm asking her if there is any recent successful programs active in breeding programs.
Okay, thank you, Mustafa. So maybe Dr. Izan, I will like to repeat the Mustafa questions. So basically, Mustafa asking how we determine the germplasm characterization by using the SSR or SNP like that, and is there any output that success by doing this kind of molecular markers, the SSR and the SNP, SNP, sorry.
Okay, so basically, okay, we have been used, okay, a lot of those marker in order to do a characterization, okay, but not maybe on the maybe it's more depend on the germplasm, okay, not the whole big germplasm that we have now in the repository spot okay, because to do that, okay, we have to do a lot of trial, okay. So this is what we want actually to achieve now, okay, with all those repository, okay, that already globally, because the repository of Spalbard is only just happened, okay, and then each of the countries give all the seed that they want to preserve in case of disaster. Okay, so the next step is that how can we use those molecular data, okay, to characterize all those accession that preserve in the germplasm. Okay, so basically, because we need to have the characterization before to know what exactly the seed for. Okay, it's like if like in my presentation just now, okay, the seed that without the characterization is not useful for us, okay, because we don't know how to use those seed, okay, so we need to do characterization, okay. So using but to do a characterization, okay, like we said just now, we have morphological, we can do molecularly, okay. So obviously molecularly is the best option to do a characterization for those in the germplasm, okay. So at least we don't need to do a field evaluation, okay, because with 750 thousand accession for germplasm, okay, so that is not really convincing to do that, right? So that is why where we can use molecular markers to do a characterization, okay. So from out of those three, maybe we can only focus after the characterization with the molecular markers, okay. So maybe we only left with 20% or 10% of the total germplasm, okay, that we call as a core collection, okay, that have all the traits that we want to preserve, okay, to be used in our breeding program, okay. So that can also, using the molecular marker, we can also detect the redundant, okay, redundant accession, redundant genotype in the germplasm, so we can minimize the number of the collection. Okay, Mustafa?
Yes, thank you so much for that clarification.
Thank you.
Yeah, thank you so much. But this is a follow-up question, like, because molecular marker and morphological marker, why molecular marker is more reliable than morphological marker in general
Okay.
diversity?
So basically, so for morphological traits, okay, is always influenced by the environmental. Okay. So if you see the plants, okay, in the field, okay, so they have uh they have a high content of maybe sugar. Okay. So but you don't know whether it is because of the genotype and or the environmental, Okay, so because for the morphological, they influenced by the environmental is too big. Okay, but if you choose the molecularly, okay, so it is because of the genotype. Okay, so remember the plant breeding. Okay, phenotype is genotype times
Yeah.
plus environment. Okay.
Thank you. Well stated. Thank you so much.
Okay, thank you, Mustafa, for your questions, and Dr. Izan for the excellent answer. And maybe next to Sabrina. Please deliver your question.
Okay, thank you for the opportunities. My name is Sabrina Rahma. My question is, I'm interested in the oil palm case study by Samsudin et al. 2025, which identified 27 SNP associated with compactness trait. Can this SNP be directly used for selection in the field, or do they still require further validation?
Yeah. For this study, the 27 markers, okay, actually we can use it directly, okay, to other population, because we develop those SNP from many populations. So we are testing now if those markers can be used out from the population that we have. Okay, for now, because we just got the data, the result, I think by last year. So and also the data is confidential with the oil palm company in Malaysia. Okay, so if you want to check, you can also go and refer to the paper. Okay, and so far I think we don't have any issue on how, because what we plan is we want to use those 27 SNP marker to act like a universal markers that associate with the trait compactness. Okay, so I think you can check those markers, okay, in the published paper. Okay.
Okay, thank you, ma'am.
Yeah. Thank you, Sabrina, for the questions. Next, the question from Andika.
Okay, thanks for the time. But sorry, can I speak in bahasa?
Boleh?
Oh ya. Terima kasih
Saya cuba ya.
moderator dan Miss Shairul. Saya ingin bertanya, saya lihat tadi di slide 14 ada beberapa software yang digunakan seperti PES, Darwin, Structure, dan lain sebagainya. Software mana yang paling mudah untuk digunakan untuk pemula, dan apakah ada yang bisa digunakan secara gratis, dan dan bagaimana data harus disiapkan untuk diinput ke software tersebut? Mungkin sekian, terima kasih.
Okay. I think Darwin and also NTSys is free available software, and for the input data, okay, on how you want to input your data is actually depend on the software. Sometimes the data needs you to have the Excel file, okay. So only Excel file from the electrophoresis gel band data, whether this marker you have the band or not. Okay, so you have to go to the specific software and check what is the input data that they are asking. Okay, or what are the file that they are needed to be as a input in the specific software. But I think antithesis is I already use antithesis during my study. So I think antithesis, I think, is the easier way. Darwin as well. Okay. So where you can just convert your Excel file into and then they can calculate all the informative content of your markers. Okay.
Thank you, Miss.
Okay, thank you, Anika, for the questions. Next questions from Franz.
Thank you for the time, for the attention presentation. My English for very fluent. May I ask my question in bahasa?
Okay.
Jadi saya tertarik Bu menggunakan molecular markers untuk tanaman lokal. Tapi di lab kampus saya tidak punya untuk mesin NGS-nya. Apakah sekiranya ada institusi yang menyediakan jasa untuk analisis seperti ini dan sekiranya harus menyiapkan apa saja?
Okay. Untuk jawab soalan tadi, sebenarnya, okay, macam kita bincang tadi, we have two types of markers. Okay, we have hybridization marker, we have a PCR amplified markers, and we have NGS marker. Okay, so those if you don't have any sequencer, okay, to do a genotyping or to do sequencing of your genome, you can start with the PCR marker. Okay, PCR based marker. Okay, so that is the most cheapest way to do molecular works. Okay, how to generate a molecular data to use molecular in your lab, to start to use molecular data.
Okay, so Franz, are you satisfied with the answer or is there any feedback?
Enough. Thank you, Miss.
And okay, maybe the last questions from Kak Michelle. So maybe I will read it because it's right down in the comments. So Dr. Izan, maybe may I ask what are the negative impacts on population genetic analysis when suboptimal markers with skewed allele distribution are used?
Okay, so if you have a suboptimal marker, okay, that have a skew allele distribution, so basically your allele is not imbalanced, okay, so meaning you have really highly numbers of allele, one highly number, and then another one is a low low frequency. Okay, so usually the low frequency will have will maybe you cannot find those low allele, the rare allele anymore in your next generation. So those are actually if you work on the skew marker. So basically for skew marker, they possess a really low PIC value. Okay, so if the majority of the population share the exact number of dominant at a locus due to the log skew distribution, okay, so the marker become useless, okay, to differentiate the parent from the uncle, okay, or the cousin or from the sibling. Okay, so that is so and if you use this marker, you will lead to the high fast positive rate in the parentage assignment, and of course when you have this problem, okay, your breeding value or your estimation of breeding value will be decreased. The accuracy of your breeding value will be decreased.
Okay, thank you, Dr. Izan. Okay, Michelle is enough. So I think that thank you for the excellent question and active participations. So with this, we can conclude our lecture and discussions. Once again, thank you, Dr. Izan, for the informative presentation and also thoughtful response for all the questions.
Thank
But
you.
before
Izan.
closing today's event, I think we would like to have a documentation session. So may I kindly ask all the participants to turn on their camera, if that possible, so that we can take a group photo. Okay, maybe we will start from the slide 1. 1, 2, 3. Okay. Next. Okay, next. Ini slide 3. Okay. Because we have done all today's agenda, and now, ladies and gentlemen, we have come to the end of today's international guest lecture. On behalf of the Faculty of Bioindustry, Agriculture, and Forestry, Brawijaya University, I would like to express our profound gratitude to Dr. Shairul Izan Ramlee for sharing her valuable knowledge, expertise, and insights with us. We would also like to thank Dr. Darmawan Saptadi and Dr. Budi Waluyo, all the students, participants for their presence and active participation throughout this event. I hope that today's lecture has enriched our knowledge and inspired future research and collaboration in the field of plant breeding, genetics, and crops improvement. Thank you once again for joining with us today. We look forward to see you at future academic events. Wassalamualaikum warahmatullahi wabarakatuh. Thank you everyone and have a wonderful